Hi, everyone, and welcome to today's webinar, Responding to SARS CoV-2 Variants such as Omicron Detection and Surveillance. My name is Jeffrey Cassell. I'm with Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots, and it's brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com/COVID-19. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them in the Ask a Question box that you see to the right and then click Submit. We'll answer as many questions as we have time for at the end of the talk. You may also submit any technical issues here if you have any trouble seeing or hearing the webinar. In addition to asking questions, we encourage you to check out our latest resources, including a new infographic on the S gene target failure that you can see on the left-hand side of your screen. And finally, we have a survey question at the bottom center if you'd like to learn more information about the TaqMan mutation panels, which you'll hear about. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education icon on the far left of your screen to obtain your credits. I'd like to now welcome our speakers for today's webinar. Vanessa Lacey is a scientist at Thermo Fisher Scientific with a Ph.D. in Biomedical Sciences from the University of California at San Diego and a bachelor's in Cellular and Molecular Biology from Cornell University. Vanessa has extensive experience in the development of IVDs and field service support, auditing of quality management systems, medical devices, and IVDs for notified bodies, and research experience in molecular biology, protein engineering, and chemical biology. In her current role, Vanessa acts as a liaison between research and development and marketing and business development to support scientists using molecular diagnostic assays and instruments. And also we have Jelena Fenenstra. She has executive diagnostic and research experience in genetics, cytogenetics, and genomics, and has a Ph.D. in experimental oncology from the Medical University of Vienna. She is the author of over 25 publications and a recipient of several awards from professional organizations, including the American Society of Hematology and the UNESCO Fellowship for Women in Science. In her current role, Jelena is responsible for driving medical and scientific affair activities in Europe, the Middle East, and Africa to help democratize molecular testing globally. Vanessa is going to kick us off today. So Vanessa, you may now begin your presentation. Thank you.
In the first half of the talk, I'll discuss how the pandemic and strategies to detect COVID-19 have evolved, diving deeper into the rapid response from Thermo Fisher. Specifically, I'll show you how the assay design of our TaqPath RT-PCR tests were built with variants in mind, and why our tests can still accurately detect SARS-CoV-2 when there's so much variability in the spike protein region. I'll then show you how the TaqPath assays can be scaled up for automated high-throughput testing using the Amplitude solution, wrapping up my section with an example from the University of Colorado and how they were able to not only meet record high volume testing demands using Amplitude, but also monitored the spread of the alpha variant by tracking the S gene target failure pattern. I'll then pass it over to Dr. Feenstra to talk to you more about mutations and variants, specifically Omicron, and how the TaqMan mutation panel and sequencing solutions can be and are being used for surveillance. So with that, I'll show you how the evolution of the pandemic has gone along with testing so far. So here's a global snapshot of the COVID-19 pandemic as of February 28th. The graph on the right depicts the confirmed number of cases recorded by each WHO region per week, with the majority of reported cases in green and yellow coming from Europe and the Americas. As of the day of this recording, there's been over 430 million cases, and heartbreakingly, over 6 million people have died. Despite all the hard work, sweat, and tears from the frontline workers, scientists, and epidemiologists all over the world, COVID-19 has not gone away. Although as we continue to understand it, adapt to it, and better educate ourselves and the public, I hope that we will reach endemic status and no longer have to suffer through these waves of hospitalization and death. What we now understand from sequencing and genotyping data is that the first clear peak you can see here in the graph in the March to June 2021 time point coincides with the emergence of the Alpha variant that first emerged from the UK. That was quickly followed by Delta, which was responsible for that second deadly wave that overtook India and quickly spread through the rest of the world in the May through December timeframe. We were then quickly overtaken by Omicron, coming out of South Africa in late 2021, and that resulted in the third and largest wave that only now is on the downslope. Incidence of Omicron in many countries has now far exceeded the incidence ever reported earlier in the pandemic. So now I'll ask you to quickly take a mental snapshot of the shape of this graph before I show you a little bit more about the situation in the US and explain why accurate testing data is so important. If you look at the red line, you can see that the situation in the US mirrors what's happening globally. I want to note that the blue histogram of daily cases and the 7-day rolling average in red represent the confirmed number of cases reported to the CDC. So what does it mean to be confirmed? Well, of course, that depends on testing. The World Health Organization defines a confirmed case as a person with laboratory confirmation of COVID-19 infection. In the published case definitions, the WHO differentiates between PCR and antigen tests. If you have a positive PCR test, that's considered a confirmed case irrespective of clinical signs and symptoms, whereas those with a positive antigen test are not considered a confirmed case unless other criteria are also met. Nucleic acid amplification tests, NAAT, also known as polymerase chain reaction tests or PCR, are considered the gold standard. They were considered the gold standard of testing very early on due to the high sensitivity, accuracy, and ability to accommodate widespread testing. Testing really is our window to understanding this virus, not only to guide patient care, but also for to understand how the pandemic is spreading. So now let's take a look at the orange line, which is the 7-day moving average of percent positivity by PCR, or the portion of laboratory performed tests that had a positive result. So why is it important to track percent positivity? According to ourworldindata.org, in a paper published by the same group in Nature Scientific Data, there's 2 reasons. 1, percent positives can track how adequately countries are testing by indicating the level of testing relative to the size of the outbreak. Countries with more widespread outbreaks need to do more testing. And second, percent positives in conjunction with data on confirmed cases helps us to understand the spread of the virus. If testing was limited, many cases would be missed. In regions with a high percent positive rate, the number of confirmed cases is likely to represent only a small fraction of the true number of infections. And where the positive rate is rising, this can suggest that the virus is actually spreading a lot faster than the growth seen in the confirmed cases in blue. So despite the record-breaking number of daily cases reported to the CDC in the most recent surge this winter, the high percent positive rate indicates that the number of confirmed cases may be vastly underestimated, even more than usual. Next slide, please. So now that we have a better understanding of why testing is so important for tracking the spread of the virus, let's take a quick walk down memory lane to see how diagnostic testing for COVID-19 has evolved thus far, and think a little bit about what we might expect in the future. In the very beginning, when the world was still in shock and we had no idea what we were dealing with, we started with symptomatic diagnoses like lung CT scans and bronchiolar lavage. These were very expensive to run at over $1,000 a test and had to be run and interpreted by experienced clinicians, and they were extremely invasive. Once we had a better idea of what we were dealing with and understood a bit more about the structure and genetic makeup of SARS CoV-2, we shifted to active disease diagnoses. This started with molecular diagnostics like PCR, which look for the genetic material inside the virus, and later included antigen testing, which uses antibodies to detect proteins on the surface of the virus. These tests were less invasive, but still not as fun as most were nasopharyngeal swabs. Testing could now be done at a central lab, and the cost per test dropped between $75 to $200. As testing demand grew, more tests became available to accommodate higher volume testing and screening. Much of the same technology was and is being used, but in a slightly different way. Specimen types became less invasive with mid-turbinate or anterior nares swabs and even saliva. Central labs adapted to accommodate higher volume testing with higher throughput solutions. And pooling and point-of-care tests emerged, including rapid antigen tests. The cost per test again dropped between $10 and $20. And where we go from there, I hope, will be more about community surveillance to identify breakouts before they spread. I imagine this will involve more widespread environmental monitoring and things like air and wastewater, which would be the least invasive sample type of all. Next slide, please. So let's look briefly at the viral dynamics of a typical symptomatic COVID-19 viral infection. The schematic on the right shows the viral load in copies per milliliter on the y-axis as a function of time after symptom onset on the x-axis. The general consensus from the literature is that live SARS CoV-2 particles have a hard time replicating in culture if the sample was collected more than 9 days after the symptoms began. This is why the viral load quickly drops in the graph. So the peak period of infectiousness and therefore highest risk of transmission is 3 days before to 8 days after symptoms began. So this is what we're looking for. There are 3 types of diagnostic tests relevant to patient management and pandemic control. One, serology test, can only provide indirect evidence of infection, only 1 to 2 weeks after the onset of symptoms, because this is when it's possible to detect the host antibodies generated in response to the pathogen or vaccine. And that part is depicted in purple and blue lines on the right side of the graph. Serology tests are thus best used for surveillance and to inform public policy, although they can't yet be used as proof of immunity. Only PCR and antigen tests can be used to diagnose an acute infection. PCR tests work by detecting the genetic material, or RNA, of the virus. RNA is amplified exponentially when short probe sequences bind to their specific target, and so very small amounts of viral genetic material can be detected. These are the most sensitive and accurate COVID-19 tests on the market, quickly being adopted as the gold standard in testing. There are hundreds of RT-PCR tests on the market right now, and although there may be slight differences in the detection limit that depends on the sample type and the instrumentation platform, they're all highly sensitive with lower limits of detection in the tens to hundreds of copies per milliliter range. Another important feature of PCR tests is the ability to amplify and detect multiple sequences simultaneously. This is done by attaching different color fluorophores to each amplified target sequence. This is what's meant by multiplexing. Where PCR tests work by amplifying and detecting sequences that encode the viral proteins, antigen tests work by detecting surface proteins, or antigens themselves. Since there's no amplification step, antigen tests are not nearly as sensitive as PCR. In the schematic, you can see the detection limit for antigen tests is at the hundreds of thousands to millions of copies per milliliter range, which means they're less likely to detect an infection if the amount of virus is low. Lower sensitivity makes antigen tests more relevant for diagnostic testing within 5 to 7 days of symptom onset, when viral loads are the highest, rather than for testing asymptomatic people or those at the beginning or tailing end of the disease when the viral loads are lower. So now that we understand a bit more about testing in general, I'd like to provide an overview of Thermo Fisher Scientific's rapid response to COVID-19. So our mission really is to enable our customers to make the world healthier, cleaner, and safer. The scale and depth of our capabilities has never been more vital. Thermo Fisher Scientific has a long history of responding rapidly in public health crises. We worked hard to make a difference, not just during the SARS CoV-2 outbreak, but also during Ebola, Zika, H1N1, and even anthrax. While we're talking about SARS CoV-2 detection kits for diagnostic and surveillance applications today, Thermo Fisher also has a hand in PPE to protect researchers and frontline workers and other scientific research tools, like those for genetic, protein, and cellular analysis, and in vaccine production and clinical trial logistics. When the first case of COVID-19 was reported in Wuhan in December 2019, researchers used our instruments to perform cryo-electron microscopy, enabling the structure of SARS CoV-2 spike protein bound to the ACE2 receptor to be published within only 2 months. Over 1,200 boxes of reagents and assays were fast-tracked so researchers could quickly develop tests. Our genetic analyzer instrument was even used to read the sequence of the viral genome, setting the roadmap for diagnostic tests. Sequence in hand, we worked rapidly to develop a multiplex real-time PCR diagnostic kit to enable clinical and public health laboratories to quickly diagnose COVID-19. We strive to be the global leader in COVID-19 testing solutions. Globally, over half of all COVID-19 testing is being performed using our technology. We rapidly scaled up our production capacity, enabling us to make over 20 million kits per week and beyond if necessary. Laboratories have been constrained with supplies of reagents, consumables, and instruments. We promise a reliable, secured supply chain to reliably meet global testing demands. We're vertically integrated as a single supplier and distributor for consumables, reagents, instruments, and software, and provide 24/7 service and support for laboratories running or looking to run COVID testing. Later in the talk, I'll share a success story from the University of Colorado and how they were able to rely on our high-volume manufacturing capabilities, secured supply chain, and focused service and support to meet unprecedented testing demands for the citizens of Colorado. The U.S. Food and Drug Administration authorized the Applied Biosystems TaqPath COVID-19 Combo Kit for emergency use authorization just days after the WHO declared COVID-19 a global pandemic. This is a highly sensitive, scalable, in vitro diagnostic solution that contains both the assays and controls needed for the real-time PCR detection of RNA from SARS CoV-2. The kit is authorized to be used on some of the most used real-time PCR instruments in the world and with a variety of sample types. Since then, we've continued to grow and enhance the TaqPath testing portfolio, enabling an uninterrupted product supply to support our customers' evolving needs. Knowing that SARS CoV-2 is not the only respiratory virus and that COVID and flu have overlapping symptoms, the following winter, we released the TaqPath COVID-19 Flu A, Flu B Combo Kit. This is a single test for SARS CoV-2, Influenza A, and Influenza B that can detect and differentiate between COVID and flu. And in Europe, we also released the TaqPath COVID-19 Flu A B RSV kit. We also offer high throughput testing options with the high throughput combo kit, which is used in conjunction with the automated amplitude solution, and also the pooling kit, 5 samples simultaneously in a single reaction, enabling more tests to be processed faster with fewer materials. The most recent additions to our testing menu use an innovative assay design with 8 targets across 3 genomic regions. The RNase P combo kit includes human sample confirmation by targeting the RNase P gene, which serves as an endogenous control to ensure sample integrity, quality, and extraction. This kit detects active infections in symptomatic patients as well as asymptomatic individuals. The Fast PCR Combo Kit is a direct-to-PCR workflow from raw saliva, enabling widespread high-frequency testing. And I'd like to draw attention to the products indicated with the exclamation point, as these ones offer the S-gene advantage, and I'll be discussing the assay design in the next part of the talk. Next slide, please. So now let's take a closer look at how the assays are designed and built with mutations in mind. The Applied Biosystems TaqPath COVID-19 diagnostic tests use a multi-target design to compensate for emerging variants and mutations. The 3 targeted protein regions, open reading frame 1AB, the spike protein, or S, and the nucleocapsid protein, N, were strategically chosen with the goal to have high specificity towards SARS-CoV-2, thereby reducing the risk of detecting any other coronaviruses that might have similar sequences. Our assay continues to remain targeted specificity to 100% of currently available complete genomes for SARS-CoV-2. In fact, we continuously monitor the literature and the databases to make sure of this. By analyzing 3 different target regions, our assay design confers built-in redundancy for mutations. If one of the probes fail, the presence of the other 2 still indicate presence of the virus. In the schematic here, you can see you would get a positive result on the left if 2 or more targets are amplified. In the top example, all 3 regions are amplified. In the bottom, that depicts what would happen if you would still get a positive result if only one of the targets was not able to generate a signal. Since region 1 and 3 are still positive, then the overall result is still positive for SARS-CoV-2. A sample is only negative if none of the viral targets are detected, and if only one is detected, that's considered inconclusive and the result should be repeated. As part of our post-market surveillance efforts, we collect, review, and analyze data on the performance of our tests, including assessing whether any emerging mutations overlap with our assay design. Currently, there is only one mutation to be aware of in relation to the TaqPath COVID-19 portfolio, and that's the deletion at position 6970 within the S gene, which is most often associated with, but not limited to, variant B11529 and B117. Since these assays are designed to detect multiple genetic targets, the overall test sensitivity should not be impacted by either variant. The S gene dropout pattern, coupled with the robust N and open reading frame 1A signal, was used as an early indicator of the B117 or alpha strain. I included a few literature references on the right showing how monitoring this S gene dropout pattern was useful during the alpha outbreak. The same phenomenon has been observed for Omicron and is currently being used as a preliminary test for the variant to be followed up by sequencing-based confirmation should the S gene fail detection. Jelena will be discussing this more with you later in her part of the presentation. So now that you understand the strategy behind our assay design, let's look at the workflow and scalability of our test. Samples are first collected from patients at a collection site. There are 6 authorized specimen types so far. The time to receive samples at the testing lab may vary. To prepare the samples, RNA is extracted from the viral particles using the MAGMAX viral pathogen 2 nucleic isolation kit. This can either be done manually or with the automated Kingfisher Flex purification system, which takes about 25 minutes to run. The qPCR reactions are then set up using the reagents and controls supplied with the TaqPath COVID-19 Combo Kit and run on a supported RT-PCR instrument. Once the thermocycling is complete, the interpretive software automatically interprets the genetic analysis results, performs QC checks against all the controls, and automatically generates a report covering each specimen. The end-to-end workflow can be completed for 94 specimens in a 96-well plate in under 3 hours, or 382 specimens in a 384-well plate in under 6.5 hours. Next slide, please. So here's an example of how you can scale your throughput by using a single Kingfisher Flex, 2 7500 fast PCR systems, and 2 full-time employees per shift over 24 hours. By staggering the workflow, you could ramp up to 2,726 results per day. And for labs that need to process even more samples and are looking for something a bit more automated, we offer the Amplitude solution. Highly automated, requiring only 4 full-time employees, Amplitude can run 1,000 to 8,000 COVID-19 samples per day. The Amplitude solution is backed by proven, innovative technology because it's compatible with the TaqPath COVID-19 high-throughput combo kit, which uses a multi-target assay design that I described earlier to compensate for emerging variants. As you may recall from the slide showing our TaqPath portfolio, the high-throughput kit is one of our products that can utilize the S-gene advantage pattern and may help identify samples infected with the 6970 deletion in the spike protein. As I stated in the beginning, we promise a secure, consistent supply of reagents and consumables, even during times when testing demands are record-breakingly high. And with that, I'm proud to show you an example of how we were able to keep all of our promises and enabled our customers at the University of Colorado at their testing lab to meet unprecedented testing demands. As they put it, they went from a boutique molecular diagnostics lab and into the fire. The gray line is a measure of the molecular diagnostics workload in the year prior to the pandemic, and you can probably guess what the red line is by the huge spike in workload in March of 2020. Their workload tripled in 3 months. Before they adopted the Amplitude solution, they were using tests and instruments from a variety of sources and were limited by the number of tests that they could run each week due to supply limitations and other allocation constraints, including expected shipments being diverted to the DoD. So without a secure supply chain, they were asked to perform over 2,000 tests per day. That was their estimate for what their throughput was going to be. With a little bit of logistical planning for the lab and hiring a few new employees, UC Health was more than happy to have a designated testing area for COVID-19 samples, given the current state of the pandemic at the time. The promise of being able to run over 4,000 tests per day and having a secured supply of over 1 million tests made the Amplitude solution the best option for them. Next slide, please. So the reason I'm so proud to share this story is because by enabling UC Health to meet these increases in testing demand, they were enabled to dramatically increase test access to the citizens of Colorado. We were able to help them remedy their previous problem with receiving materials by providing palletized shipments of 20,000 or 40,000 test kits. In fact, we even provided an extra inventory of 50,000 kits so that they could have them on hand if the need arose. The only unexpected problem was that they had so many kits they didn't know where to store them, and so they had to use the hallway. Overstock was a much more preferable issue than understock, and so the end result is that they were able to exceed their initial testing demand for 2,000 tests per day. On the histogram on the bottom right, you can see the number of RNA tests used every 2 weeks, beginning in March through June 2021. On the very first bar on the left, from March, you can see that the total number of tests dramatically rose from around 4,000 every 2 weeks to over 30,000. Just to remind you, when they were doing their planning and they were trying to predict what the testing demand might be, they thought they'd be doing only 4,000 every 2 weeks. And so I'm happy that amplitude worked out so well for them because the demand was even higher than what they expected, peaking at over 30,000. So aside from being able to meet the volume of test demands, they were using the high throughput kit that offers the S gene advantage, and so they were also able to monitor the spread of the alpha variant based on this unique result pattern. They never encountered this S gene pattern until early February, and at that time, the UK was also monitoring the same pattern. And when they reported that the 6970 deletion was present in over 99% of the sequence of the sequence dropout samples, and the European CDC also recommended using multi-target PCR assays to investigate further, UC Health then decided to send over 2,000 of their samples out for sequencing, and lo and behold, over 99% of them were indeed B117. The histogram on the right shows all of the COVID positive samples coming from amplitude between February and June from the alpha wave, and categorizes each result based on the S gene pattern. You can see the ones in orange that exhibited the S gene pattern form a peak that mirrors exactly what we now know as the alpha wave. And with that, I'd like to conclude part one of the presentation. PCR testing demand is now higher than ever with the current surge in Omicron. Thermo Fisher Scientific has been at the forefront of the pandemic and is a global leader in COVID-19 testing solutions. The TaqPath multiplex assay design targets 3 regions of SARS CoV-2 to compensate for emerging mutations and variants. The TaqPath workflow is adaptable to high volume testing needs with the Amplitude solution. The S dropout pattern along with the Amplitude solution, were useful for meeting high volume testing needs at the University of Colorado. And with that, I'd like to pass it over to Jelena for the second part of the talk.
Thank you so much, Vanessa. So my name is Jelena Feenstra, and today I would like to talk about the emergence of different SARS CoV-2 variants, including Omicron, why they are important, what are their clinical implications, and what surveillance solutions can be applied for their detection, discrimination, and identification in patient samples. So since the detection of the original strain of SARS-CoV-2 in Wuhan in December 2019, the global transmission of the virus has led to significant viral evolution. And while all viruses mutate, RNA viruses mutate at high rates like SARS-CoV-2, and under selective pressures, the new variants are emerging. The SARS-CoV-2 virus was estimated to acquire 1 to 2 mutations per month, but throughout the pandemic, several SARS-CoV-2 variants emerged, which contained higher number of mutations than scientists anticipated. And this phylogenetic tree here shows the evolution of SARS-CoV-2 from the initial Wuhan strain identified in December 2019, which is shown here in the center. And the circulating variants are classified into lineages, and these concentric circles represent the time throughout the pandemic. And you can see with each month that was passing by, the picture was getting more and more complex with more variants emerging. In some periods during the pandemic, there were high numbers of different variants circulating. For example, if we look at 2020, we can see several variants of concern that were circulating at the same time, such as alpha, beta, and gamma, to be later replaced by the Delta variant in 2020, and most recently by Omicron at the end of 2021, shown here in red. So what are the implications of these variants? So first of all, the mutations that these variants carry might offer the virus an advantage for it to spread more rapidly and, through the population with increased human-to-human transmission. Second, they may be associated with higher viral loads and affect the disease severity and lead to worse clinical outcomes. And of course, some mutation might render the virus more resistant to treatments such as antibody therapies, or they can lead to escape of vaccine immunity or immunity acquired by a previous infection with a different viral variant. And of course, some of the mutations in the variant may impact the performance of diagnostic tests which are used to detect the coronavirus. And based on these key characteristics and the risk that they pose to public health, the variants are classified into these four categories, variants being monitored, variants of interest, variants of concern, and variants of high consequence. Now, so far, no variant has been designated as variant of high consequence, but there have been several variants of concern. And let's look at those in a bit more detail. So currently, according to the US CDC, there are two variants which are considered to be variants of concern, and these are Omicron and Delta. And there was a number of other variants, and there you see them. They are currently classified as variants being monitored in this gray box, and some of them marked in red, such as Alpha, Beta, Gamma, and Epsilon, were actually previously having the status of variants of concern during 2020 and 2021. And if you look at the two graphs on your right, you can see the timeline of the pandemic, and you can see that there were several variants that were very dominant throughout this period. So, for example, first such variant was Alpha, which emerged in the UK in autumn of 2020, and you can see there was a period of time when this variant was accounting for the majority of COVID-19 cases in the US, marked here on the lower right graph in red. However, then the Delta variant emerged in summer of 2021, and you can see that in green that it very sort of over time overtook all the other variants that were circulating and became the dominant variant in the US, but also worldwide. And then as the Alpha, Beta, Gamma, and other variants were not circulating and not accounting for a high proportion of cases in September 2021, they have not been considered anymore as variants of concern and have been given this variant being monitored status. And then you see in November 2021, the Omicron variant emerged, and really in record time, as shown in this graph in purple, it took over the Delta variant and became the dominant variant in the US. And again, this graph represents the United States situation, but very similar picture could be seen for many other countries worldwide. Now, if we look a little bit more closely at the mutation profiles of some of these main variants of concern, it's important to understand that the SARS-CoV-2 virus has the so-called spike or S protein on its surface, which binds to the ACE2 receptor on the cells of the respiratory tract and enables the virus to enter the cells and infect them. And as it is on the surface of the virus, the S protein is also the target of most of the vaccines that have been developed for SARS-CoV-2. Now, mutations in SARS-CoV-2 variants of concern affect the S gene, and in this figure you can see in purple all mutations in the S gene in the Delta, Omicron, and Alpha variants of concern. And what is immediately visible is that there is a significantly higher number of mutations in the Omicron variant compared to, for example, Alpha or Delta, and there were 30 in total. And this high number of mutations was one of the main concerns when Omicron was first detected, as it was expected that if a protein undergoes so many mutations, it might more efficiently evade the immune response induced by vaccine or previous infections. And what you can also appreciate from this figure is that there are certain mutations which these variants of concern have in common, and we've marked them here with these red arrows. And I just want to point out one of them, which is, for example, the deletion 6970, which is an important mutation that was found in Alpha and later on in Omicron variant, but was not present in the Delta variant of concern. And some of these mutations are referred to as mutations of concern, also because later on functional studies demonstrated that they have an impact on the viral properties. So in the next couple of slides, I would like to focus a little bit more on the Omicron variant. So the Omicron variant was first detected in November 2021 in South Africa, and by January of 2022, it was present in almost every country worldwide. And the emergence of this variant was responsible for the latest wave of SARS CoV-2 infections, and the incidence was really above everything else that was earlier reported during the pandemic. So you can see on this upper graph the situation in different WHO regions, and you can see that the peak of the infections was much higher than previously seen during the pandemic. But what was also observed, if you look at the graph below, showing situation in the UK, that the peak was also very sharp, so the number of infections rose significantly and then also dropped rather quickly. And, you know, if we look at the features of this Omicron variant, now the first thing that was very clear is that it had a growth advantage over the Delta variant, that there was an increased risk of infection, and that in addition, it did have immune escape properties in terms of increased risk of reinfection and potential to escape vaccine-induced immunity. And interestingly, the Omicron variant was found to localize more in the upper respiratory tract with potentially reduced ability to infect the lung, and thus also that led to reduced risk of hospitalization in infected individuals. Now, of course, with this high number of mutations that we've seen that S protein of the Omicron variant has, there was a potential effect on some of the monoclonal antibodies that were applied in treatment of patients with COVID-19, while, for example, diagnostic tests were mostly not affected by this variant. And, you know, as Vanessa already explained, and the three-target design of the SARS-CoV-2 assay and the fact that the S gene target does not amplify if this deletion 6970 is present in the virus can really offer an advantage in terms that the same assay that is used for the detection of the virus can provide an indication for the presence of variants that carry this deletion 6970, and one of these variants is Omicron. And in fact, the group of Tulio de Oliveira, who actually first reported on the Omicron variant, later on published an article in Science where they explained how this S gene target or failure or S gene dropout, how we refer to it, can be used to track the spread of the Omicron variant. US FDA and also other agencies stated that the pattern of this S gene dropout may be a signal for the Omicron variant, while the sensitivity of the test is not impacted, but of course the variant would need to be confirmed by additional methods such as sequencing or genotyping. And in fact, for example, the United Kingdom public health agencies have used the S gene target failure to track the spread of variants carrying this deletion 6970 throughout the pandemic, actually at a national level. And these are some graphs that were published by the UK government. And I just want to show you, for example, in the upper graph, how actually at the end of 2020, the laboratories noticed that there were some samples starting to show up with the S gene dropout. They are shown here in purple, and that was actually the time when the alpha variant emerged, and this is how they detected it, and they tracked its spread. And later on, sometime you see here around May 2021, they suddenly saw that there are samples that are showing amplification of the S gene. So there was no more S gene dropout. This is the green curve. And this is actually the time when the Delta variant emerged and how they could use now the amplification of the S gene to track the Delta variant spread in the UK. And similarly, they've used this exact same feature of the TaqPath assay also when the Omicron variant emerged at the end of 2021. You see again the purple curve and how they could detect samples with the S gene dropout. And clearly, you know, should the next variant replacing Omicron not carry this deletion 6970, this feature of the TaqPath assay could again be useful in this sort of terms of surveillance. So, of course, I would also like to mention some of the sublineages of Omicron. And in this graph, you can see them listed. So there are several sublineages. They are referred to as BA.1, BA.1.1, BA.2, and BA.3. And you can see in this upper image some differences in mutation profiles between these different lineages. And for example, the BA.2 variant was first reported actually in Denmark, because this is one of the countries that was first to drop all of these public health measures against the coronavirus. And you can see on this slide or on your lower right on the image that the BA.2 variant very quickly became the dominant variant actually in Denmark at the beginning of 2022. And this picture was not only observed in Denmark, it's the same, for example, in South Africa, India, and quite a few other countries. What we know about this BA.2 sublineage is that it has an increased growth rate compared to BA.1 Omicron, which was the main circulating lineage previously, and a study published by Danish researchers where there were the most cases already showed that there is a higher secondary attack rate among contacts. It's important to point out that this particular sublineage does not carry this deletion 6970, so it is actually S gene target positive. And now in the next part of my talk, I would like to present some of the surveillance solutions for detection and discrimination of SARS-CoV-2 variants. Now, of course, the gold standard for SARS-CoV-2 detection is RT-PCR on RNA isolated from nasopharyngeal swab samples or other sample types. And once we know that the virus is present in a sample, there are methods for variant and mutation identification. And that can be done either, for example, with Sanger sequencing to confirm specific mutations, or with RT-PCR genotyping assays that can provide a very rapid option to detect a certain mutation of concern. And the advantage of using a targeted mutation panel, of course, is that the results can be obtained more quickly and on the same instrument which is used to detect the presence of the virus. Now, of course, we also have the, let's say, what is considered more classic surveillance methodologies, which would be the sequencing solution, which can be performed either on certain parts of the genome, such as the S gene, which we know undergoes a lot of mutations, or next generation sequencing could be used to sequence the entire viral genome and potentially identify novel variants. And I would like to go now in a little bit more detail about each of these specific technologies and how one could implement them in their specific settings. So, for example, the TaqMan SARS-CoV-2 mutation panel offers a menu of more than 60 verified real-time PCR genotyping assays. And we know that, some of the mutations in these variants are shared between the variants, and some are very specific for a particular variant of concern. So laboratories are very flexible, and they can decide on their own what kind of panel they want to build for variant detection. The advantages are their results can be obtained very quickly and using the same instrumentation as for detection of the presence of the virus. And this approach also offers some flexibility in terms when the new variants emerge or when some variants stop circulating, then these parts of the panel can be, of course, edited. And now how to discriminate the variants using a mutation panel. So here depicted on your right is an allelic discrimination plot, which is just an example to show if you run one genotyping assay, what would the results look like? And you could see here that, for example, each dot represents one sample. And for example, the red dots would represent samples in which the assay detects the mutation. And the blue cluster dots would be the samples that would be wild type for this particular position that we are looking at. And here I've just listed some of the assays we have available for the Omicron or for the Delta variant. And of course, the labs can choose how many assays they would like to run to be able to discriminate between the Omicron and Delta positive samples. And if you want to know more about how mutation panel was used to track the Omicron variant spread in the US, there is this publication which resulted from a collaborative effect between the NIH, Helix, Rosalind, Thermo Fisher Scientific, and other partners. And within this study, a 4-assay panel was evaluated for detection and differentiation of Delta and Omicron variants of concern. You can see in this table here the results of the study demonstrated the performance of the genotyping approach for detection of these variants when compared to whole genome sequencing. And in this particular panel, they have chosen 1 assay specific for the Delta variant and 3 assays specific for the Omicron variant. And the results show that the genotyping approach had a very good agreement of 100% PPA and 99% MPA as compared to whole genome sequencing. And the real-time tracker to support the rapid genotyping program for SARS-CoV-2 in the United States was developed by Rosalind. It can be accessed at the link provided below. And here on this slide, you can just see the screenshot of the information that this tracker is depicting and the currently circulating SARS-CoV-2 variants across the US. The additional significance of this tracker is that if we start seeing a trend where these undetermined cases that don't fall into alpha or that don't fall, into Delta or Omicron start to increase, it means we are looking at a potentially new variant showing up that we would then need to sequence and identify further. And of course, sequencing is a method of choice for identification of new variants. The next generation sequencing can be used for surveillance of the full viral genome, and the Ion Ampli-6 SARS-CoV-2 Insight Research Assay covers 99.7% of the viral genome and all potential serotypes, and it enables complete sequencing of the SARS-CoV-2 genome even in the low viral load samples while offering excellent performance as the virus mutates. This assay can be used on the Ion Chef and Ion Gene Studio S5 series systems, but also on the fully automated Ion Torrent Genexus integrated sequencer, which offers a 1-day turnaround time. Now, of course, individual amplicons can be confirmed using Sanger sequencing, which we know is the gold standard for sequencing short stretches of the viral genome and detection of mutations. It is cost-effective, and the protocols and primer sequencing sets are available for surveillance either of the full length of the S gene, which undergoes highest number of mutations, or for confirmation of SARS-CoV-2 variants. And of course, labs can select low to high throughput options for sample to result workflow in 1 day. And in my last slide, I would just like to summarize by saying that SARS-CoV-2 continues to evolve and will likely become endemic with new variants emerging in the future. We know that testing continues to play a critical role in patient management, also in containing the spread, in tracking current variants, and early identification of new variants. Thermo Fisher Scientific offers a comprehensive menu of testing solutions for SARS-CoV-2, including the ultra-high throughput options to deal with the surges, as we recently witnessed with Omicron, as well as for surveillance of emerging variants. So for more information, please do visit www.thermofisher.com/COVID-19. And I believe it is now time for questions.
Okay, thank you, Vanessa and Jelena, for that wonderful presentation. We're going to start now the live Q&A portion of the webinar. If you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far right of your screen, and we'll answer as many of your questions as we have time for. So let's go ahead and get started. It looks like we have 2 questions coming in for Vanessa. So given the differences in sensitivity between antigen and PCR tests, when does it make sense to use either technology? And as a follow-up, you've talked a lot about lab-based PCR tests and high-volume testing. Does Thermo Fisher offer any point-of-care test solutions? So Vanessa?
Yeah, thank you. So in general, antigen tests are less sensitive, but they're a lot quicker to run and cheaper to run. This is why the CDC recommends following up any negative antigen test with a PCR for confirmation, especially if you're symptomatic. Antigen tests are cheaper and offer the promise of a rapid turnaround time, often within minutes, and so they're suitable to use when prevalence is really high and you need to quickly identify symptomatic individuals who are the most likely at risk of transmitting the virus to others. So due to their much higher sensitivity, PCR tests are ideal in any situation where you need to have very high confidence that your first result is accurate. There's actually 2 main classes of PCR tests. So there's the lab-based PCR that we discussed mostly in the talk. And those use well-established lab processes that are run by trained technicians. And although it only takes a couple hours to run the actual test, you also have to take into consideration the time that it takes to transport the samples to and from the lab, making the turnaround time a little bit longer, but still usually within a day. Lab-based PCR tests are the easiest to scale up, and they're ideal for high-volume processing, supporting local and national level testing efforts, such as in schools or in corporations. And rapid PCR, so these are more point of care. So this uses newer technology, and that allows it to be performed at the site of sample collection. Rapid PCR tests are ideal for evaluating high-risk congregate settings, such as emergency rooms, clinics, hospitals, and nursing homes. And we do offer the Acula SARS CoV-2 test, and the features of that include getting results within just 30 minutes. It's a streamlined process with a CLIA-waived workflow. It has a simplified sample collection supporting nasal swabs and nasal mid-turbinate swabs. And another benefit is that it can be stored at room temperature, so you don't need refrigeration. And since it is based on PCR, the results are highly accurate. The percent positive agreement was 95.8% to 100%, and negative percent agreement was 100% in both prospective and retrospective clinical studies. And it's also highly sensitive with the LOD less than 500 RNA NAAT detectable units per milliliter using a reference panel. So yeah, good question. Thank you.
All right. Okay, great. Thanks so much, Vanessa. So this next question I think is for Jelena. How many assays are needed in order to detect a variant of concern using the genotyping approach?
Yeah, so as I said during my talk, every variant will carry mutations which are, specific for that particular variant, and as well as those mutations that might also be present in other variants. So it's very important that at least one assay that a lab selects to run is specific for that variant, but how many in total the lab chooses to do is really depends on the lab and the current situation in the region they are in. And as I demonstrated during my talk, for example, one approach to differentiate between Delta and Omicron was to pick for assays, but for example, some other labs might want to use at least 2 assays per variant, or at least 3 assays per variant, and so on. And of course, it also depends on how many variants are circulating at the same time. So for example, in 2020, when we had all of these at the same time, then of course the labs were picking a little bit bigger panels. And again, I would like to stress that this flexibility that the laboratory has to remove assays from the panel and add new assays as new variants start to emerge is also very, very important. And we found that the labs have found this very useful in their practice.
Great. Okay, thank you so much. I think we've got time for one more question. So let's see here. So now that the Omicron variant is the main circulating variant, can S-gene target failure still be useful in the context of surveillance? So Jelena, this looks like a question for you again.
Yeah, sure. So the answer is absolutely, and in fact, we already see that it is very useful because, as I've shown you, in some countries like Denmark and so on, there has been the spread of the BA2 Omicron sublineage that still in the US accounts for a small proportion of the cases. But for example, this variant doesn't carry the deletion 6970, which means it would have the S gene amplified, and the labs would very easily be able to track the BA2 variant spread because they could see the difference between positive samples with the S gene dropout and those without the S gene dropout. So, we have seen the value of this feature of the TagPath assay really being used by many labs worldwide. And of course, the exact variant would need to be then confirmed by additional testing, but at least the lab would know how to focus these efforts really on the samples that, that we already know what you could expect, which variant is in it.
Okay, great. Thank you so much. All right, so thank you again, Jelena and Vanessa, for your time today in discussing Thermo Fisher's Omicron response and solutions for detection and surveillance. We would also like to thank LabRoots, our sponsor, and Thermo Fisher Scientific for underwriting today's educational webcast. Before you go, I'd like to thank you, the audience, for joining us today and for your interesting questions. We apologize that we didn't get to all of them today. The webcast can be viewed on demand, so LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. And so until the next time, have a good day and goodbye. Thank you for joining us.
Thank you so much.